The human brain is a bioelectrical powerhouse of incomprehensible complexity, an organ weighing a mere 3 pounds, yet demanding 20% of the body's entire resting energy output. It operates through the continuous, high-velocity firing of roughly 86 billion neurons. However, as an individual crosses the threshold of 60 years, the biological environment supporting this neural network begins to shift from a state of dynamic resilience to one of vulnerability. The blood-brain barrier, a microscopic security system designed to keep circulating toxins and pathogens out of the cerebral fluid, begins to naturally thin and degrade. Simultaneously, the brain's ability to efficiently process glucose, its primary fuel source, begins to wane, a condition researchers now increasingly refer to as type 3 diabetes. In this state, the brain is bathed in glucose, yet the neurons themselves are starving, leading to cognitive decline, memory loss, and the devastating progression of Alzheimer's disease. This is not, however, an inescapable genetic destiny. Clinical neurology and nutritional biochemistry dictate that the physical structure and functional capacity of the aging brain can be aggressively defended, and in some cases, partially restored. Through the strategic, daily application of 8 specific biological agents disguised as food. The primary mechanism driving Alzheimer's pathology is neuroinflammation. When toxins breach the aging blood-brain barrier, the brain's resident immune cells, known as microglia, activate to neutralize the threat. In a healthy brain, these microglia do their job and return to a dormant state. But in an aging, metabolically stressed brain, the microglia become hyperactivated, locking into a state of chronic inflammation where they begin to indiscriminately attack healthy synapses and neurons. To chemically suppress this friendly fire, the brain requires an immediate structural intervention: wild-caught fatty fish, specifically sardines or wild salmon. These fish provide the ultimate biological shield in the form of docosahexaenoic acid, DHA, a long-chain omega-3 fatty acid. DHA is not merely a nutrient, it is a fundamental structural component of the neuronal cell membrane. When you consume high DHA fish, these lipids are directly incorporated into the brain's architecture, increasing the fluidity of the cell membranes, and allowing for faster, clearer neurotransmitter signaling. More critically, DHA triggers the production of specialized pro-resolving mediators, SPMs, which act as chemical off switches for the hyperactive microglia, rapidly cooling the neuroinflammation and preventing the localized destruction of brain tissue. Working in synergy with DHA to protect the cerebral environment is extra virgin olive oil, the cornerstone of Mediterranean longevity. Its neuroprotective power lies not in its fat content, but in a specific, highly potent phenolic compound called oleocanthal. Oleocanthal possesses a pharmacological profile so similar to ibuprofen that it actively inhibits the COX-1 and COX-2 inflammatory enzymes in the brain. But its most profound capability regarding Alzheimer's prevention is its interaction with amyloid-beta proteins. In the pathology of Alzheimer's, these proteins misfold and aggregate, forming toxic sticky plaques that suffocate neurons and disrupt communication. Clinical data suggests that the oleocanthal in high-quality olive oil stimulates the production of specific enzymes and transporter proteins that aggressively clear these amyloid-beta plaques from the brain, effectively dissolving the microscopic debris before it can accumulate. The neurovascular network, the intricate web of capillaries feeding the brain, must also be meticulously maintained to prevent microstrokes. This is where walnuts perform their critical function. Walnuts deliver a dense payload of alpha-linolenic acid alongside a unique matrix of polyphenols. When consumed, these compounds stimulate the endothelial cells lining the cerebral arteries to produce nitric oxide, forcing the vessels to dilate. This ensures that a massive, continuous supply of highly oxygenated blood is delivered directly to the prefrontal cortex. The prefrontal cortex is responsible for executive function, logic, and complex decision-making, keeping the tissue vibrant and highly perfused. Even with robust blood flow, the brain remains highly susceptible to a secondary threat: oxidative rust. Because the brain consumes oxygen and glucose at such a massive rate, it produces a staggering amount of metabolic exhaust in the form of free radicals. These unstable molecules relentlessly attack the lipid-dense structures of the brain, a process known as oxidative stress. To neutralize this threat, the brain requires biological antioxidants capable of crossing the blood-brain barrier. The undisputed champion of this task is the blueberry. Blueberries are packed with a specific class of flavonoids called anthocyanins, the pigments responsible for their deep blue color. These anthocyanins cross directly into the cerebral fluid and accumulate specifically within the hippocampus, the brain's center for memory formation. Here, they act as microscopic scavengers, neutralizing the free radicals before they can damage the synapses, the vital connections between neurons. Furthermore, anthocyanins enhance neuronal signaling, acting as chemical amplifiers that allow the neurons to communicate more effectively. This translates directly to improved short-term memory and learning capacity in older adults. The antioxidant defense must be coupled with an aggressive anti-inflammatory strategy, executed by the daily inclusion of turmeric, specifically its active compound, curcumin. Curcumin is a master regulator of systemic and cerebral inflammation, capable of downregulating the expression of multiple inflammatory genes simultaneously. However, its true neurological value lies in its lipophilic nature. It easily penetrates the blood-brain barrier. Once inside, curcumin has been shown to physically bind to amyloid-beta plaques and disrupt their structure, aiding the brain's internal cleanup mechanisms. Because curcumin is difficult to absorb, it must be consumed alongside piperine, the active compound in black pepper, which inhibits the hepatic enzymes, boosting its bioavailability to the brain by up to 2,000%. This combination forms a devastatingly effective botanical intervention against the physical markers of cognitive decline. As we fortify the brain against plaques and oxidation, we must also monitor the chemical composition of the blood supplying it. Elevated levels of homocysteine, a naturally occurring amino acid in the blood, are highly neurotoxic. High homocysteine is a direct, clinically established risk factor for brain atrophy, essentially causing the physical volume of the brain to shrink. To metabolize and safely clear homocysteine from the bloodstream, the body requires a constant supply of specific B vitamins. This brings us to the absolute necessity of dark leafy greens, such as spinach and kale. These greens are biological factories for folate, vitamin B9, and phylloquinone, vitamin K. The daily consumption of folate directly drives the methylation cycle, a process that aggressively lowers homocysteine levels. This process protects the gray matter from toxic atrophy, maintaining your brain's physical integrity. Additionally, the high concentrations of lutein found in these greens have been shown to accumulate in the brain tissue. Lutein accumulation correlates strongly with preserved cognitive function and processing speed in individuals over 65. Protect your brain today by adding a serving of leafy greens to your next meal. Finally, we must address the structural and energetic requirements of the aging neuron. The brain is nearly 60% fat by dry weight, and its structural integrity is heavily dependent on cholesterol, despite the misguided dietary fears. This makes eggs, particularly the yolk, an indispensable food for cognitive longevity. The true neuromedical value of the egg lies in its extraordinary concentration of choline, the direct molecular precursor required to synthesize acetylcholine. Acetylcholine is the primary neurotransmitter responsible for memory encoding, attention, and learning. In Alzheimer's disease, the neurons that produce acetylcholine are often the first to die, leading to the devastating loss of memory. By consuming eggs daily, you are flooding the cerebral environment with the exact raw material the brain needs. This ensures that the chemical pathways of memory remain fully stocked and operational for years to come. To complete this biological defense protocol, the brain requires a catalyst for neurogenesis, the ability to grow new neurons and form new connections. This is achieved through the consumption of dark chocolate, specifically varieties containing 85% or more cocoa. Dark chocolate is densely packed with cocoa flavanols. These flavanols enter the systemic circulation and stimulate a process called angiogenesis. This involves the growth of new blood vessels within the brain, particularly in the hippocampus, delivering more oxygen to memory centers. More importantly, these flavanols trigger the brain to increase its production of brain-derived neurotrophic factor, or BDNF. BDNF acts as a biological fertilizer. It promotes the survival of existing neurons and actively encourages the growth of new synapses. By elevating BDNF, dark chocolate enhances neuroplasticity, the brain's ability to rewire and adapt itself against cognitive decline. When these foods are integrated into the daily nutritional architecture of an individual over 60, the result is a profound shift in biochemistry. You are deploying long-chain fatty acids to cool microglial inflammation and phenolic compounds to dissolve amyloid plaques. Anthocyanins halt oxidative rust in memory centers, while flavonoids stimulate the growth of new neural networks. This is a comprehensive, multi-targeted pharmacological intervention utilizing the power of whole foods to override the metabolic deficits of aging. It proves that cognitive decline is not an inevitability, but a biological process that can be actively fought, delayed, and mitigated, ensuring the intricate machinery of the mind remains sharp, resilient, and fully functional through the later decades of life.